Hi everybody, today we shall discuss some of the questions related to DNA replication from the chapter Molecular Basis of Inheritance. You can have a look at the diagram that is related to DNA replication as given here in the slide. So let us enter into the questions related to it. Coming to the first question. Semi-conservative model for DNA replication was proposed by Watson and Crick. Hershey and Chase gave the ultimate proof for DNA as the genetic material by carrying out transduction experiments in E. coli by using the bacteriophage T2. Jacob and Moyad proposed Rotanon concept to explain the regulation of gene expression in E. coli. Messelson and Stahl experimentally proved semi-conservative replication of DNA in E. coli, but the model was first given by Watson and Crick while giving the model for DNA structure. The fourth option must be the correct one. Let us see the answer. Fourth one. Semi-conservative model for DNA replication was proposed by Watson and Crick. Hershey and Chase gave authentic proof for DNA as genetic material. Jacob and Monod gave operon concept to explain regulation of gene expression in E. coli. Messelson and Stahl experimentally showed semi-conservative replication of DNA in E. coli. Coming to the next question. So it is related to DNA replication. It's the matching. A is the template strand. That are a template DNA strand on which the lagging daughter DNA strand is formed. So template A3. B is replication fork. So we know that at each replication bubble there is formation of two replication forks. So B is the replication fork 1. A3, B1. C. What is C? C is the lagging strand. 4. C4. D is the leading strand or leading daughter DNA strand. C lagging daughter DNA strand. So A3, B1, C4 and D2. First option must be the correct one. Let us see the answer. First option. A is the template DNA strand on which the lagging daughter DNA strand is formed. B is replication fork. C is lagging strand with Okazaki fragments. D is the leading daughter DNA strand. Coming to the next question. So, related to Messelson and Stahl experiment. So initially, the DNA of E. coli or E. coli cells DNA is labeled totally with N50 ensured by growing for several generations E. coli cells in FT N15S4Cl containing medium. They ensure that DNA of all the bacteria will be having N15, that is heavy nitrogen. So DNA will be heavy. That's why the DNA of that bacteria is isolated and subjected to cesium chloride density gradient centrifugation. So it's very clear in the background that the DNA is suspended almost at the lower level of the centrifuge tube. So that shows that is a heavy DNA. So it's very clear here it is heavy DNA. So after transferring to N14 containing medium, means N14H4Cl containing medium, after one generation or 20 minutes time, so there is a formation of only hybrid DNA. Hybrid DNA means one strand with the N15, this one is with the N15, this one with the N14. So they have got intermediate density. That's why both the DNA molecules are suspended somewhere in the middle, because the DNA is hybrid DNA. Again, after another generation, after second generation, there is a formation of 4 DNA molecules. Right? Out of 4, 2 will be having only lighter DNA strands N14, and 2 will be having each one strand N15, another one N14. So half of the DNA is hybrid DNA. So this one is the hybrid DNA. Here it is the And the remaining of this one and this one is the lighter DNA. The hybrid and lighter DNA of the second generation is formed in 1:1 ratio. Here only hybrid DNA, first generation. No lighter DNA. No heavy DNA. Right? Here lighter and hybrid are formed in 1:1 ratio of the second generation. So this experiment was carried out by Messelson and Stahl. The first option must be the correct one. Let us see the answer. The experiment was carried out by Messelson and Stahl in 1958. Next one. Statement 1, Statement 2. Semi-conservative mode of replication of DNA was shown in eukaryotes by Taylor and his co-workers. Absolutely true. Right? Statement 1 is correct. So, Taylor and co-workers showed the semi-conservative mode of replication of DNA in the chromosomes of a eukaryote known as Vicia faba or broad bean or faba bean. N15 isotope was used by Taylor. No, not N15 isotope, it is thymidine, right? Radioactive thymidine was used, not N15. So statement 2 is wrong. So saying N15 isotope was used by Taylor to demonstrate semi-conservative replication of DNA in eukaryotes is wrong. Statement 1 is correct, statement 2 is incorrect. 1 is correct, 2 is incorrect. The first option must be the correct one. Taylor and his co-workers used radioactive thymidine to demonstrate semi-conservative replication of DNA in chromosomes of eukaryote Vicia faba, faba bean of Fabaceae, can also be called as broad bean. Coming to the next question. So, E. coli cells that were grown in N15H4Cl containing medium transferred to normal N14H4Cl containing medium and allowed to grow for 80 minutes. 80 minutes means E. coli cells divide once in every 20 minutes or 18 to 20 minutes. For 80 minutes means there is formation of 4 generations of divisions. 20 minutes first generation, 40 minutes second generation is completed, 60 minutes third generation is completed, 80 minutes fourth generation is completed. Right? So after first generation only, as we have seen earlier, only hybrid DNA is formed. Right? Second generation, hybrid and lighter DNA 1:1 ratio. Right? So the third generation, if you say that the third generation, there is a formation of 8 molecules of DNA. Out of 8 molecules of DNA, 2 molecules will be hybrid. Remaining 6 molecules will be lighter. So 6 are lighter and 2 are hybrid means 6:2, that is the 3:1. Right? That is 60 minutes. After 80 minutes, there is a formation of, say, if you say that there are a formation of 16 molecules, 10 to the power of 4, 16 molecules. Out of 16 molecules, 2 will be always the hybrid DNA. Remaining 14 will be lighter DNA. Lighter 14 and hybrid 2, 14:2 means 7:1. So third option must be the correct one. This is third option, is third one. After 20 minutes or first generation or replication or division, only hybrid DNA is formed, 2 molecules. After 40 minutes or second generation or replication, one portion is light, 2 molecules, one portion is hybrid DNA, 2 molecules formed. Hence the ratio is 1:1. After 60 minutes or third generation or replication, 3 portions is light, 6 molecules, and one portion, 2 molecules is hybrid DNA. Ratio is 3:1. After 80 minutes or fourth generation of replication, 7 portions is light, 14 molecules, and one portion, 2 molecules is hybrid DNA. And the ratio is 7:1. So this question was asked in the textbook. What happens after 80 minutes, right? Or after fourth generation of replication was given in the book. No answer was given. So that we have to find out. This is the answer for that particular question. Coming to the next question. Assertion driven type. Replication of E. coli genomic DNA is completed in about 18 to 19 minutes. In your book it is given as 18 minutes, but actually it takes slightly more than 19 minutes. Right? Assertion is correct. The polymerization rate of DNA polymerase of E. coli is 2000 base pairs per second. Yes. 2000 base pairs per second. In E. coli, only one RE site is present in its genomic DNA or nuclear DNA. At RE site, there is formation of one replication bubble. One replication bubble has got two replication forks. At each replication fork, one set of enzymes for DNA replication is present. So there are two sets of enzymes for DNA replication. So for their activity, the two sets of DNA replication enzymes are able to finish complete length of DNA of E. coli genome in about 18 to 90 minutes. We know that it has got 4.6 into 10 to the power of 6 base pairs. That's the genomic DNA. So, assertion is correct, reason is also correct, and the reason explains the assertion. A and R are correct. R correctly explains the assertion. Answer must be the two. One answer. Here is the explanation. You can also read out. In E. coli, there is only one RE site in its nuclear DNA. It forms two replication forks at its replication bubble. Two sets of enzymes are involved in replication, one at each replication fork in both the directions. Each set polymerizes 2000 base pairs per second and two sets 4000 base pairs per second. A total of 4000 into 60 into 19 base pairs are polymerized. Polymerized. In the 19 base pairs are polymerized. It is almost 4.6 into 10 to the power of 6 base pairs. Here 19 is the time that is taken. Right? Polymerized. And total number of base pairs 4.6 into 10 to the power of 6 base pairs. It is almost 4.6. So that is the case. It is the 4.6 into the power of 6 base pairs. Coming to the next question. DNA replication in E. coli is semi-discontinuous and bidirectional. So continuous now, semi-discontinuous or semi-continuous. Unidirectional now, bidirectional. Discontinuous now, bidirectional now. It is semi-discontinuous and bidirectional. Third option, semi-discontinuous, bidirectional. Yes, third option must be correct one. Fourth option, semi-discontinuous, unidirectional. Wrong. Third option must be the correct one. The semi-discontinuous because on one template DNA strand is continuous, another template DNA strand discontinuous. And two replication forks are found. One replication fork moves towards forward direction, another replication fork moves towards backward direction. So bidirectional. So third option must be the correct one. Let us see the answer. DNA replication E. coli is semi-discontinuous because at a replication fork on template strand with a 3' 5' polarity it is continuous, and on template DNA strand with a 5' 3' polarity it is discontinuous. At a RE side there is the formation of two replication forks which are moved simultaneously in opposite directions by two sets of replication enzyme protein complexes. Coming to the next question. Identify the correct statement. Identify the correct statement. Everything is given here. Identify the correct statement. Energetically, DNA replication is a very expensive process. Absolutely true. Energetically, DNA replication is a very expensive process. A is correct. Deoxyribonucleoside triphosphates not only serve as substrates for DNA replication but also provide energy for myelinization. This is also true. Deoxyribonucleoside triphosphates not only serve as substrates because the monophosphate part, nucleoside monophosphate part is incorporated into the DNA and the pyrophosphates are released during the breakage of the bond, energy release. That energy helps in the formation of new bonds, that is phosphodiester bonds. Right? So B is also correct. DNA replication is not only very fast but also occurs with high degree of accuracy. Absolutely true because there is also proofreading and proofreading enzymes are also there. Any wrong nitrogen base or nucleotide is inserted, that is removed and replaced with the right nucleotide nitrogen base during replication. This is also correct. Only DNA dependent DNA polymerase is involved in DNA replication. Now, several enzymes are involved. So, main one is the DNA polymerase or DNA dependent DNA polymerase. That is usually DNA polymerase 3, right? So, for proofreading and correction, there is also involvement of DNA polymerase 1, right? Other DNA polymerases. There is also involvement of helicase, topoisomerase, right? So, several enzymes are involved and primase is also involved. For DNA molecules, so D is wrong. For DNA molecules of any length, the two strands of DNA are separated in its entire length at a given stage. No, only a part of the DNA is separated, not the entire length at a given stage. Goes on gradually being separated. One side there is replication, other side separation takes place. So E is also wrong. A, B and C are correct. Second option must be the correct one. So only at a small region the two strands of DNA are separated at a given stage. Not only DNA dependent DNA polymerases, but also several other enzymes such as helicases, DNA ligases, topoisomerases or gyrases, and primerases are also involved in DNA replication. Coming to the next question. Choose the incorrect statement. Incorrect one you have to choose. DNA polymerases on their own, DNA polymerases on their own can initiate the process of DNA replication now. DNA polymerases on their own cannot initiate DNA replication because they require a pre-existing nucleotide over the template. A pre-existing primers must be present. Otherwise, they cannot function or carry out. So they require a pre-existing nucleotide or primer over the DNA template. Then only they can attach later the successive nucleotides one after another to the primer, whose sequence is complementary to the sequence of template DNA strand. So first is incorrect statement. First itself is incorrect. Let us see other three are correct or incorrect. DNA replication is initiated at a region named origin of replication. This is correct statement. Vector used in recombinant DNA technology must possess site for origin of replication. Absolutely true. Otherwise, the gene carried by the vector cannot be replicated. So DNA replication occurs in 5' 3' direction. Absolutely true. So DNA replication or RNA synthesis or DNA synthesis or daughter DNA strand formation or new RNA formation and template DNA strand always takes place in 5' 3' direction. 2, 3, 4 are correct statements. First one is the incorrect statement. So the answer is 1. DNA polymerases on their own cannot initiate the process of DNA replication. For DNA replication initiation, first at 80 rich regions of RE, they vary at 80 rich regions. So 13 base pair regions, 9 base pair regions are there. So that they are 80 rich regions of the RE. The two strands of DNA are separated to a sharp stretch by a portion to form a replication bubble having two replication forks. So that 9 base pairs regions it is separated. Two replication forks are formed. So at each replication fork, helicase enzyme is loaded with the help of protein. So this helicase enzyme helps in breaking the adjoining bonds to move the replication fork. So by two other regions where replication has to be carried out. So that is the third one. Single stranded binding proteins are bound to the melted strands to prevent reannealing. Yes. So after the breakage of hydrogen bonds, immediately SSBP, single strand binding proteins are bound to the template DNA strand to prevent the reannealing of the separated parental DNA strand. RNA primers are formed by primase RNA polymerase on both the strands as the DNA polymerases cannot polymerize the nucleotides unless a pre-existing short sequence of nucleotides are present over templates. Already we have discussed this one. Right? The incorrect statement is the first one. DNA polymerases also do proofreading and later primers are removed and fragments of DNA of lagging strand are attached by DNA ligase. Coming to the next question. DNA ligase enzyme, what it does? So after the removal of primers of the fragments of DNA ligase enzyme attaches the fragments of DNA of lagging strand by forming phosphodiester bonds. So that is DNA ligase enzyme. So let us see the options. Forms phosphodiester bonds between discontinuously formed DNA fragments on template strand with 5 prime 3 prime polarity. Yes, template strand with 5 prime 3 prime polarity, the lagging strand is formed, and lagging strand has got discontinuously formed DNA fragments which are known as Okazaki fragments. Each Okazaki fragment has got one primer. The primers have to be removed during proofreading by DNA polymerase 1. After that, then only the between fragments phosphodiester bonds are formed by DNA ligase. First one is correct. Forms phosphodiester bonds between discontinuously formed DNA fragments on template strand with 3 prime 5 prime polarity. No. On template strand with the 3 prime 5 prime polarity, there is continuous formation of the daughter DNA strand which is one leading strand. Fragments are not formed. So second option is incorrect. Forms phosphodiester bonds during leading daughter DNA strand formation. No, this is wrong. Right? This is wrong. This is also wrong. Forms hydrogen bonds. No, it forms only phosphodiester bonds. First option must be the correct one. Let us see the answer. DNA ligase forms phosphodiester bonds between fragments of lagging DNA strand, that is Okazaki fragments, or discontinuously formed daughter DNA strands fragments after removal of primers and replacing ribonucleotides with deoxyribonucleotides by DNA polymerase 1 by its exonuclease activity. Only after the removal of primers and replacing the ribonucleotides with the deoxyribonucleotides, there is formation of phosphodiester bonds by DNA ligase enzyme between Okazaki fragments. So, please like, comment, and subscribe. Thank you.